Great. Thanks, Dr. Weiss. So just to briefly introduce one of our endowed lecture series from infectious disease, the Hoffman-Ratzin Lectureship was established in 2005 by the grateful trainees of Dr. Thomas Hoffman and Dr. Kenneth Ratzin, who led the infectious disease training programs at Jackson Memorial Hospital, and Mount Sinai, starting in the 1970s. Dr. Hoffman received his medical degree at the University of Pennsylvania before completing a fellowship in infectious diseases at the University of Rochester. And Dr. Hoffman was chief of infectious diseases at the University of Miami from the 1970s all the way to 1998. And Dr. Kenneth Ratzin completed his medical degree at Harvard Medical School before residency training at Columbia Presbyterian Hospital and ID fellowship at New England Medical Center in Boston, and was chief of infectious diseases at the Miami VA Medical Center in 1972 before moving to Mount Sinai Hospital in Miami Beach, where he served as chief of infectious diseases until June of 2020. And Dr. Ratzin served in many leadership roles, including chairman of Department of Medicine, director of medical services, and program director at Mount Sinai for the internal medicine residency program. Both were widely recognized through the community and set very high standards for their trainees, which I can attest to as a trainee of Dr. Ratzin. This visiting lectureship was established to bring ID leaders from around the country to the University of Miami Miller School of Medicine to honor the tradition of scholarship set by these outstanding leaders. And I will say the Division of Infectious Diseases is thrilled to virtually host Dr. Robert Bonomo, a world-renowned leader in the field of infectious disease and antimicrobial resistance, as this year's Hoffman-Ratzin Grand Rounds speaker.
Awesome. And I'm Andres Rodriguez. I'm one of the chief residents, and I'm happy to speak and introduce our guest speaker today, Dr. Robert Bonomo. He's a distinguished university professor of medicine, pharmacology, molecular biology, and microbiology, biochemistry and proteomics, and bioinformatics at Case Western Reserve University School of Medicine. He also serves as Associate Chief of Staff of Academic Affairs at the Louis Stokes Cleveland VA Medical Center. He's an elected fellow of IDSA, American Academy of Microbiology, the Association of American Physicians, the European Society of Clinical Microbiology, and has been appointed PCORI as a representative of the IDSA. Today, he will delve into the renaissance of beta-lactamase inhibitors, and it is my distinct pleasure to welcome Robert Bonomo to Medicine Grand Rounds.
Thank you very, very much, Dr. Rodriguez, for that very nice introduction. It's a real honor to be here today, and I am thrilled to be able to share with my colleagues some things that we've learned over the years. First, you know, if I'm trying to move my slide, a special thanks, you know, to Lillian. You know, she and I go way back, and we've had the privilege of working together. And I had the great honor today to speak with Dr. Shulka and Dr. Dobelki Lewis this morning. It was a real pleasure to meet them again. And, you know, all my buddies over the years. You know, no one can go through science or medicine being isolated. The privileges that we have is not only to be partakers of a great profession, but also to know so many people who have been so generous with their time over the years, and I'm very grateful for all of them. So let's start. I have to tell you the truth. When I was used to do ID before, I used to have tons of slides about the importance of antimicrobial resistance and show them, and then I stopped. I stopped doing that, and the reason I stopped doing that is because when this publication came out, you don't need to say anything more. By 2050, increases in antimicrobial resistance will be responsible for 300 million deaths and have a total of more than 100 trillion US dollars. That's an amazing toll on society. And I think as we begin to evolve and begin to look at what antimicrobial resistance is around the world, we can see that in many of these under-resourced areas, going to be extremely challenged by the impact of antimicrobial resistance. Over the years, my lab has been focusing on beta-lactamases. These are bacterial enzymes that function to inactivate beta-lactam antibiotics. And there's 4 different flavors of them. There are 4 classes. Class A, C, and D are serine enzymes. and class B are the sort of notorious, if you will, metallo-beta-lactamases. And when you look at their mechanism, to understand how to overcome beta-lactamases, you have to understand what the mechanism is. And many times, infectious disease fellows, when they see these compounds, they start to get a little nervous. And, a lot of clinicians, when they see compounds like this and arrows and all that stuff, get nervous. But it's really very, very simple. What a beta-lactamase does is, by using a strategically positioned water molecule, both for the serine enzymes and for the metalloenzymes, these strategically positioned water molecules allow the breaking of this bond. When that bond is broken, the beta-lactam can no longer be an effective agent to inactivate a penicillin binding protein. Our lab has studied beta-lactamase inhibitors. And one of the things I tell the fellows in the lab, and I visit the lab every morning, is that beta-lactamase inhibitors live and die at the bench. And the more we know about beta-lactamase inhibitors as they're coming out into the clinical arena, or even before they come into the clinical arena, our patients will benefit from that. And they're complicated. They make, there are many intermediates as, forms the Michaelis complex, and then you form an acyl enzyme. And these different pathways occur. You can form tautomers, different shapes, different quantities. And it's all very, very complicated, but the end result is that these beta-lactamase inhibitors, by inactivating the beta-lactamase, preserve the utility of or give a second life, if you will, to the partner beta-lactam. We are going to have lots of choices. This is a beautiful painting from Cezanne that, many, many choices out there that we will have. And right now, everybody's used the old beta-lactamase inhibitor. Sometimes I call them the first generation, clavulanic acid, sulbactam, and tazo. Now on the market, we have avibactam, relibactam, and vaborbactam, combined with either a carbapenem or a cephalosporin. These are compounds that are here to stay. And on the very, very near horizon, we have a deluge, if you will, of compounds that will be coming down the pike that, with different chemical scaffolds, will be able to use in the clinical arena. And there's one antibiotic that I think really needs a beta-lactamase inhibitor, and that's cefiderocol. This is a molecule and a class by itself, but these are ones we have that are coming down the pike. And I'll touch upon all of them and give you a brief update. But, the most difficult science, I think, is that all these compounds are going to be good. But the most difficult science, I think, will be how to use these the best way possible. And also to design diagnostic assays and clinical trials to get to that answer is not going to be trivial. So we have challenges ahead. The first new beta-lactamase inhibitor was avibactam. And in some ways, avibactam or Taz-Avi is the gold standard, because everything that's going to be talked about is going to be compared to avibactam. And I had the good fortune early in 2005 or 2006, sounds like years ago, that we did the due diligence on this compound when it was being developed in France. So I know about Avi for many, many years. And one of the interesting things chemically is that avibactam resembles the cephalosporin that it's paired with right over here. And that's why it's a very, very good inhibitor. And I can just envision what people were thinking when they developed this compound, when they tested it first against a KPC2 producing Klebsiella pneumonia, carbapenem-resistant Klebsiella pneumonia, when the ceftazidime MIC was greater than 512. You add a little bit of avibactam, and that MIC falls like a rock down to 0.25. Totally untreatable, totally treatable. And I bet, people in the lab, when they were talking with first describing this, probably went to the pub that night and, and celebrated their good fortune. And, you can see how potent this compound was against a lot of, even OXA48, which is a real problem in Europe, not so much in the United States, but a very good inhibitor against OXA48. We also figured out early on that it was not going to be good against metallo-beta-lactamases and not going to be good against certain class Ds. So we knew early and this is a very nice data that was summarized in and the basis for this lecture when we wrote this paper. Sarah Jaws was a PhD student in my lab at the time, and she did a very nice job summarizing this data. How does avibactam work? Well, Professor Foco Vandenacker at Case Western Reserve really did some very nice work on this compound. He was able to look at the crystal structure, and we discovered that this took a chair-shaped conformation in the active site. So now, when you show crystal structures to people, to docs, they always get nervous because they think they don't understand it. But if you follow the wiggles like I do, you can see how this is sort of bending and then bending again. So the chair-shaped conformation, and it slowly desulfates, which is an important thing to know in another context, that it makes sure that the enzyme can only be inhibited once. So what is the real world application? Well, Professor David Van Dyne, transplant ID doc at UNC, put together a very nice study where we compared ceftazidime-avibactam to colistin against CRE infected individuals. And this was so remarkable data. I'm so excited about this data, is that in patients treated with ceftazidime-avibactam versus colistin, the all-cause hospital mortality at 30 days was 9% for the Taz-Avi group and 32% for the colistin group. That is significant, statistically significant, and all-cause propensity-adjusted mortality was significantly decreased with avibactam compared to colistin. And when you look at the graphs that were generated in that paper, this is the hospital death rate of the colistin group. This is the hospital death rate of the Taz-Avi group. There's a huge difference. And when you look at colistin being combined with other drugs, it still didn't do as well as Taz-Avi alone. So, which, I used to like to, joke about this, but one good drug is better than two bad ones. And that's what Taz-Avi has done in this particular arena. There's been lots of other studies, many retrospective studies. Some of you may also have participated in some of these. The Reclaim trial, where Taz-Avi compared to meropenem, both had very, very good efficacy. Recapture 1 and 2, Taz-Avi versus Dory in complicated UTI. Reprise, both complicated UTI and complicated intra-abdominal infection versus best available therapy, 82% efficacy. The Reprove trial, which is a nosocomial infection, which also showed that Taz-Avi was comparable or maybe a little bit better than even meropenem. And there's lots of retrospective case series. But unfortunately, with antibiotic resistance, there ain't no free lunch. And a number of years ago, about a decade ago, in the lab, Christina Papwalis and I were doing some site-directed mutagenesis on the KPC enzyme in order to understand how resistance to antibiotics emerged. And we made this very unfortunate discovery that changing some amino acids in the omega loop of the enzyme created Taz-Avi resistance. That was only relegated to the scientific journal. It wasn't a clinical problem at the time, but unfortunately, things happened in the clinic, and it was only a period of time where Ryan Shields and his group from the University of Pittsburgh described Taz-Avi resistant isolates that were emerging while patients were being treated. We had done work in this and were anticipating that this change in the certain part of the enzyme called the omega loop was responsible for this. And with Brad Spellberg, we wrote this editorial that was in CID, and we sort of mimicked the Jaws thing. You're going to need a bigger boat, because we thought that this was going to be a real problem. And it actually became a real problem. So in Cleveland, we established collaborations with people in Bristol, England, also work that was done in our lab, and work that was done at the University of Miami in Oxford, Ohio, looking at different aspects of the KPC enzyme. And we really tried to understand this as best we could. We looked at expression levels. There was no change. We looked at the conformation of the protein, and we found out that using NMR, that the conformation of the protein was drastically changed, about 3%. We did molecular modeling, and we showed that the hydrogen bond disorder was very evident. And we did some heat mapping, thermal stability testing, and we found out that D179 was not stable. We did further experiments with mass spectrometry, and we showed that avibactam took a while for avibactam to get on the enzyme. So there was some resistance to avibactam, but the real hooker was the resistance to ceftazidime. When Professor van den Akker tried to do crystallography, he could not locate very clearly the part of the enzyme that was disordered as a result of a mutation in the omega loop. Professor Jim Spencer in England did a very important structure of a deacylation deficient enzyme. We knew that ceftazidime could fit. We didn't know what would the effect of this deacylation mutation in the enzyme, but it allowed us to trap ceftazidime in the active site. And with that, we went back to the old literature. And in the old literature, Shahryar Mobasheri figured out a long time ago that in certain compounds, it could be the compound itself that accelerates its own release from the enzyme. And with Brian Soyket's help, and starting this, we studied whether this would be something that the enzyme itself did, or the compound was responsible for its own catalysis. And to cut a long story short, we postulated that ceftazidime itself is the culprit for its own resistance in the active site of KPC that's resistant to ceftazidime avi. We went back and tested a oxacefm, moxilactam, to see whether our theory was right, whether ceftazidime itself was the culprit here. And sure enough, when you test moxilactam against ceftazidime avibactam resistant strains, moxilactam resistance does not go up, while ceftazidime avibactam resistance does. So there may be some truth to this hypothesis. We'll find out. There's still more studies that we're doing, but maybe for certain cephalosporins, this oxacefm class could be a good one to revisit to help us preserve the activity of these compounds. And we've done some molecular modeling further to show that. What about meropenem-vaborbactam? This is a very interesting drug. Vaborbactam is an oxaboronate. It's a boronic acid derivative, but instead of having a free boron, it's part of a ring with the oxygen atom right next to it. Very, very potent inhibitor of KPC, a very good inhibitor of ESBLs and of AMP-C enzymes. And the trial of meropenem-vaborbactam versus piperacillin-tazobactam, this was a phase 3 trial enrolling patients called Tango 1 that went from November of 2014 to April of 2016. And in Tango 1, they showed that the primary endpoint and overall success was 98+% with Mero-Vabor versus 94 compared to Pip-Tazo. In Tango 2, which was against carbapenem-resistant Enterobacteriaceae, they stopped this trial early because Mero-Vabor was very effective against CRE, and it was associated with increased clinical cure, as was ceftazidime avibactam, with less nephrotoxicity compared to best available therapy. But just like Taz-Avi, Merovaber can become resistant. And when you start losing porins, which is a story we know in infectious diseases, because when you have Pseudomonas aeruginosa that becomes carbapenem resistant, the most likely explanation in the United States is a porin loss as opposed to metallo-beta-lactamase. But as you begin to lose more porins in Klebsiella pneumonia, that this was studied on, nice summary by Dr. Christina Papp-Wallace in Infectious Disease Clinics of North America, the more porins you lose, the more resistant you become to meropenem vaborbactam, which is a shame because it's also a very good antibiotic. And one of our fellows in our lab, Dr. Mohammed Yasmin, did a very nice study that was presented at ID Week, where lots of Klebsiella, even a long time ago, that were KPC producing, were Merovaber resistant because of these porin channels. Imipenem relibactam, a very, very important drug to have on your formulary. Relibactam, the inhibitor, it inhibits KPC very, very well and restores susceptibility. This was an important study that we were very fortunate to be sponsored by Merck on this. This formed the basis for using ImiRel against ceftazidime avibactam resistant strains. ImiRel is able to overcome KPC2 mutants that are resistant to ceftazidime avibactam, which is something that I think some hospital formularies may want to keep in mind, particularly if when they use Taz-Avi, if resistance emerges in the clinic. But nevertheless, what's the data to show that in a pivotal phase 3 trial, imipenem cilastatin with relibactam was much better when it was compared to colistin and imipenem in patients treated with ImiRel. The clinical response at day 28 was significantly better than when it compared with less toxicity when it compared to colistin and imipenem. Studies done in published in CID Restore IMI2 showed that all the parameters when it comes to 28-day all-cause mortality and overall clinical response was much more favorable for patients treated with imipenem relibactam versus the comparators. So, and even against Acinetobacter, Enterobacter complex, et cetera, against all these criminals, I like to call them, in the drug-resistant world, relibactam was compared favorably to best available therapy. Now, Dr. Abu, in a very nice poster that was presented at ID Week, summarized, and some of you in the audience are probably on this poster, summarized the clinical experience. I had the privilege of speaking with Rosanna again this morning. It was great. And, where they looked at the real world experience of ImiRel against Pseudomonas aeruginosa at the major medical center. And again, very, very favorable activity. But like Merovaber, like Taz-Avi, resistance to imipenem relibactam emerges. And just like in Merovaber, it's the loss of the porin channels that gives you the very high MICs to imipenem relibactam. Interestingly, in Pseudomonas, efflux does not have an impact on ImiRel. It's more the presence of the OPRD mutant that drives resistance to ImiRel. There are many, many stars in the sky that we have to look for. So what about the new ones that are coming down the pike? So many of you that went to ECMID heard about Nacubactam. Sometimes I joke it's an inhibitor in search of a beta-lactam. This was a drug developed by Roche a number of years ago. The big difference here is that it has this amino-ethoxy side chain, this amino-ethoxy side chain compared to avibactam. What Nacubactam does, which is very interesting, it also binds to penicillin binding proteins like PBB2. In binding to PBB2, it also inactivates the the beta-lactamase. So you get a double effect when you use nacubactam. You not only inhibit the beta-lactamase, but you also inactivate a penicillin binding protein. And we showed this in our lab. If you take Taz-Avi-resistant Klebsiella pneumonia that have those bad mutations that cause Taz-Avi resistance, nacubactam by itself compared to Taz-Avi MIC of 1 versus an MIC of 8. So obviously, when you combine it with meropenem, you get even lower MICs. And since these Taz-Avi resistant strains, they become hypersusceptible to meropenem, you get this phenomenon. But even nacubactam by itself has its own activity. And I think in the future, people will want to develop these sort of novel inhibitors that have both this sort of activity. I know everybody here has used ceftolozane, tazobactam. Ceftolozane is actually a very, very smart cephalosporin. When you start breaking down to each of the components of this compound, it's a big compound. And the reason it's so effective is that it's big and it doesn't fit into the active site of the AMP-C beta-lactamase. But it has lots of parts to it that enhance activity, confirm beta-lactamase stability, enhance anti-pseudomonal activity. So each part of this compound has a really special role. And it, as a result of its good activity, it was found to be non-inferior to meropenem in the ASPECT complicated intra-abdominal infection trial. Against ASPECT complicated UTI, was superior to levofloxacin. Toltezo did better against ESBLs. Against nosocomial pneumonia and ventilator-associated pneumonia, was non-inferior to meropenem, and overall clinical success was 74%. And, unfortunately, like I told you, the Taz-Avi story and the IMI-REL story and the Mero-Weber story, there's a price that's paid with all these compounds. And unfortunately, if you look at the AMP-C beta-lactamase of Pseudomonas, when you put in certain mutations, you get resistance to ceftolozane, tazobactam. So these enzymes have within themselves the ability to confer tol-tazo resistance, just like an ESBL emerges in the clinic. These enzymes emerge in Pseudomonas aeruginosa that convert tol-tazo resistance. And very, very early on, working with Professor Shozeb Haidar at the University College of London, we were able to do some very fancy metadynamics, which is, studying how the protein moves in space over a period of time and how these free energy wells develop. We were able to show that there was a hidden pocket that appeared in the AMP-C of Pseudomonas aeruginosa that permitted the enzyme to capture the molecule in the active site and to hydrolyze the cephalosporin. So by making the enzyme more flexible, by making the enzyme more open, it's able to capture ceftolozane in the active site and inactivate. There are other beta-lactamase inhibitors, and in some ways, this is a masterpiece of medicinal chemistry that we will have so many options. One that's coming on that's in phase 3 trials is cefepime-zidibactam. This represents a new chemical class by targeting 2 different PBBs. It acts synergistically against Pseudomonas and Acinetobacter. So this will be a very good anti-Pseudomonal drug and an anti-Acinetobacter drug. And a number of years ago, working with Professor Moya in Spain, we were able to show that zidibactam bound to a lot of penicillin, bound to PBB2 with almost as good as a beta-lactam antibiotic bound to PBB2, and converted all these living cells into sphericals by inactivating PBB2. With Christina Papwalis, we did a microbiological and chemical and structural survey of different beta-lactamases inactivated by zidibactam. And now this compound is also able to inactivate penicillin binding proteins. We were able to study this in Pseudomonas aeruginosa. And this is why, in coming down the pike, cefepime Zidibactam, which I think many of you guys will think about, its application against hospital-acquired pneumonia, ventilator-associated pneumonia, UTI, and bloodstream infections. It's stable to metallo-beta-lactamases, so which is going to mean it's going to have some role there, and also has good activity against extremely drug-resistant Pseudomonas aeruginosa, carbapenem-resistant Acinetobacter, and even Stenotrophomonas. So right now, the trial is ongoing of cefepime and Zidibactam, and we're all anticipating the results. Venadarex is going to be coming out with a cefepime taniborbactam combination, and I think Dr. Abu and I were at the conference where a lot of this was talked about for the first time at an ID week, and cefepime taniborbactam, when you compare it to Tazavi, Toltezo, has very, very nice MICs, nice shift to the left in the distribution, very, very potent compound, also against metallo-beta-lactamases. It was shown at Bristol by one of our colleagues, Jim Spencer, that it forms a nice complex inactivating both the metallo and serine beta-lactamases. So this will be used against NDM1 producing strains. And this is just a cartoon showing how taniborbactam, another oxyboronate, is able to coordinate to the 2 zinc ions and keep those 2 zinc ions from breaking down the water molecule that's important in the activating the water molecule that's important in the hydrolysis of of cephalosporin or a carbapenem. So this is a very smart compound. Inactivates the 2 metal ions, which will allow us to treat NDM1, VIM, and producing metallo-beta-lactamase strains. In the certain one, this was a trial. This was talked about at ID week. Patients, this was for acute pyelo and acute urinary tract infection. Patients received treatment for up to 14 days. If they were bacteremic, 661 patients, 66% comprised the MITT component, 42% had acute pyelo, 58% had a UTI, many were aged over 65, and the drug cefepime taniborbactam had a clinical and microbiologic response rate of 88 and 87, 88.7 and 86%, and did extremely well. So I think this was a very good trial for them, and they compared very favorably to meropenem. Another drug that you'll be hearing a lot about, particularly if you have an Acinetobacter problem in your hospital, will be the combination sulbactam, darolobactam. This was a combination and a beta-lactam inhibitor, darolobactam, combined with another beta-lactamase inhibitor, sulbactam. But the trick here is that sulbactam in Acinetobacter inactivates the penicillin binding protein 3 and 1. And a number of years ago, we worked with Entasis and showed that you could use the sulbactam, darolobactam, and lower MIC significantly. These were isolates that we got from the Walter Reed Army Medical Center. The beta-lactamase inhibitor effectively inhibited the class C and the class D beta-lactamase. The ATTACK trial, which there's going to be a supplement coming out in the CID shortly on sulbactam, darolobactam. In this trial, all patients received imipenem cilastatin in addition to either sulbactam, darolobactam, or colistin. And in the mortality, in the sulbactam, darolobactam against very, very hard to treat Acinetobacter was 19% versus 33, almost 33% in the colistin arm. So very, very favorable comparison between sulbactam, darolobactam, and colistin. Keep in mind, even though the imipenem cilastatin was given to both arms of that study, so we have to see how that's going to shake out. But The summary that most clinicians know is that in the Sol-Dur, in the Sol-Bactam had lower mortality, higher clinical cure, significantly greater microbiologic and favorable outcome than those treated with colistin. And all the data is here. If you actually want to see this, and I think this data is going to be published very soon. So I'll just skip that. Another important inhibitor that is coming out is N-metazobactam. When we started first working with the drug, it was called AAI 101, and sometimes I was worried about it, so I used to say ay. But anyway, I was wrong. N-metazobactam is actually very, very effective, better than piperacillin tazobactam. Against ESBLs that we had. And, we could have predicted, why the Marino trial was going to be so successful. When you use pip-tazo against certain ESBLs, you don't get reduction in MICs like you think you should. Some SHV ESBLs are really resistant to tazobactam and inactivation. The MICs are high. And metazobactam compared much more favorably. And it's probably the addition of this small methyl group on this triazole ring that enhances the activity, because it becomes a positively charged molecule. And as a zwitterion, this is the negatively charged part of the molecule. This is the positively charged part of the molecule. It gets into bacteria a lot better and a lot faster than tazobactam, which doesn't have that. So it's, in some ways, it's a very smart addition. The biochemical characterization of N-metazobactam, it's not as good as avibactam against KPC, but not too bad, but not so bad. So there could be a hidden benefit to this drug. We were able to show that an ESBL like CTXM15, which is the part of ST131 E. coli that is all over the world in terms of its threat. When you treat N-metazobactam binds to CTXM15, we saw something we never ever seen before, but there was a formation of a cross-link across the active site between two amino acids. And this was an alternative mechanism for beta-lactamase inhibition, which I think probably also happens in the bacterial cell. And this is probably contributes to the reason why N-metazobactam, when it's combined with cefepime, would be so effective. The QPX compound, this is an ultra broad spectrum inhibitor, in the words of the company, that works against both serine and metallo-beta-lactamases and has really very good activity. It's and sometimes there's been some discussion whether QPX should be allowed to be marketed on its own, where you, the clinician, picks the beta-lactam partner. We could debate that if there's time at the end of the talk. I'd love to hear how you guys feel about being able to pick your own partner beta-lactam. I think it would be a real interesting discussion. The drug is also being paired with an oral beta-lactam, so it's going to be both IV and PO. And since it inhibits so many different beta-lactamase, it got the term ultra broad spectrum, and some of its activity, it's also extremely highly soluble, also very, very highly bioavailable. So it's going to be absorbed very well, mixes with water nicely, so you don't have problems with that. And even is going to have activity against Acinetobacter and NDM producing metallo-beta-lactamases. So for those of you who are interested, we summarize here some of the structural studies that show, that the catalytic water molecule is covalently bound by QPX. And just like the story with taniborbactam, because QPX is a cyclic oxyboronate, we have this relationship, and the drug is going to be able to be manufactured and given to patients orally. So what do you do when you don't have these drugs on the market and you need you have a metallo-beta-lactamase producing strain that's a threat to imipenem, that imirel won't work against, merovaber won't work against, ceftaz-avi won't work against, and all the other old beta-lactamase inhibitors won't work. And in some very, very nice work that was championed by Dr. Maria Mojica, who is now a senior instructor at Case Western Reserve. This was work she did when she was a pre-doc. She was, by understanding the basic mechanism, we were able to divine a strategy where we use ceftazidime, avibactam, and aztreonam together as a combination to treat a child, a renal transplant child who was about 60 days of constant bacteremia with stenotrophomonas multifilia, failed colistin treatment, failed minocycline treatment, the introduction of Tazavi and aztreonam. Both are FDA-approved drugs, even though the combination is still under investigation. We're able to cure this child of his renal transplant. And, this was the one time, not the one time, but we all take pleasure from, having to really help people. And I could remember when, we saved this renal transplant, even though we were in another city, far away from Columbus, not far away, but 2-hour drive from Columbus, Ohio. I think I actually came to tears, when we discovered that this child's renal transplant was saved. So we went on to further, test this in the lab. Can you reduce CFUs in an animal infection model? Can you do hollow fiber? And we did all the appropriate thing. And most recently, Tom Lodis has just published a phase 1 study of Tazavi and aztreonam and has demonstrated its safety. I have a slide on that. I just didn't add it for today, but I could send people the PDF. This is one of the compounds we're working on in our lab. This is also a bornate, MB076. We hope that since it keeps the mice alive, that eventually when we test it in humans with another combination other than cefepime, it will also work as well. We work with another group in South America to develop inhibitors for metallo-beta-lactamases that resemble penicillin, and these, we have a derivative now that seems very promising. So, in conclusion, there's lots of beta-lactam, beta-lactamase inhibitors out there. We're going to have a lot of choices. And, what are the ones that are going to promise to be really good? Well, clearly, sulbactam, darulobactam is going to play a big role against Acinetobacter. I'm certain of it. I think some of the ones that have activity against metallo-beta-lactamases like cefepime, taniborbactam is going to be an important drug, and cefepime, zidibactam is going to have some role there, as will the QPX compound, et cetera. And I think, we have to figure out the best way to use cefiderocol. I think that'll be a discussion that I'd also like to have here. But, what I can say in closing is that as we, bring these drugs on our formulary, I think we have to use them wisely. And this is why antibiotic stewardship programs are really critical here. Because, Tazav, Toltezo, Tazavi, Imirel, they're not inferior to standard therapies for easy to treat infections. If Piptazo works, we should stick with Piptazo. If, Amsulbactam works, stick with Amsulbactam. You don't need to go beyond it, but it's against your multi-drug resistant organism, that's when the most benefit occurs from ceftolozane, tazobactam, Tazavi, Imirel, and merovaber. And I just summarized for the fellows if they want to know and keep these slides, what are the different mechanisms of resistance, not to forget about them against these novel agents, because they are very important, particularly with merovaber. Be worried about OXO48 like bearing strains, because merovaber will not work there. So there are challenges ahead. I always joke, finding the right beta-lactam for your beta-lactamase inhibitor is like getting married. If you got the right partner, you're golden for the rest of your life. And knowing a good idea when you hear one is also very important. I think we're going to have a lot of new tools. We're going to have to study the biological correlates of infection and make sure we understand it. These things are still very, very elusive to us, but we're applying newer and newer technologies, cryo-electron microscopy, atomic force microscopy, et cetera, and new techniques to help us understand why these bugs become resistant. And this was my favorite book when I was in training, and also still now. No Time to Lose. This was Peter Pio's work, who was masterful in the elucidation of the Ebola virus and Marburg virus and HIV/AIDS in Africa, and ran the United Nations program there. We have no time to lose. And instead of studying deadly viruses, some of us are studying deadly beta-lactamases. And we should always look for forward-looking science to benefit our patients that is based on sound theory, and which should be our mandate. We need to marry good hypothesis testing at the bench and in the clinic. So with those remarks, I think we have a couple of minutes for questions. I thank you so much, and thank you, Dr. Weiss, for this invitation, and thank you for this unique honor.
Thank you, Dr. Bonomo. That was a beautiful grand rounds. We're grateful that you were able to review everything so clearly for us. And we're also grateful we do have great antibiotic stewardship programs, both at the University of Miami and at Jackson, where we all work. And we couldn't survive, or more importantly, our patients wouldn't, without their expertise and our expert infectious disease consultants. One, perhaps for the more generalists in the audience, perhaps you can leave us with some words of wisdom with regard to how can we prevent the further, it seems you say like you get a marriage partner, it's one for life sometimes. How can we assure that we stay married to this person forever? In other words, which antibiotic would, or what, the way in which we use them, what words can you give us to be sure that we don't induce more resistance coming into the future?
That's an excellent question. And in the past 3 days this week, I've been asked that very same question, because when you talk about these new drugs and you see how resistance emerges, we don't want to be there. We don't want to be there. And we don't want to take a great, powerful drug like Tazavi or Imirel and waste it. And so I think there are two answers to that we should be really, really careful how we employ our antibiotic stewardship programs and listen to what they say when they say, you really need to de-escalate. And, I think that's a critical thing. And the other thing that's really important here is that we really don't understand evolution. And we describe evolution very well. We say that this thing went from this to this, which is a description. We don't always have a good explanation for why that happened. Like, what are the fundamental forces of nature? What are the fundamental forces in bacteria that create this evolutionary process of drug resistance? People blame it on things like mismatch repair, and they blame it on too much excess use. But fundamentally, the driving forces of evolution are still mysterious. They're still mysterious. And, one of my great pulmonary friends, he said to me, Robert, if we knew what problems we would get into, we wouldn't do it. We would avoid it. And unfortunately, one of the hardest things to change in life is behavior. We tend to be repetitive, and the mistakes we made years ago sometimes are the mistakes we continue to make. And, evolution is blind to that. Evolution is blind to that. And, I think, the more we understand about these very fundamental scientific truths that make things change, is the better we'll get at that. But while we don't understand them, I think controlling their use, having good stewardship programs, having very good, people out there, like in the Division of Infectious Diseases that you have a stellar program there. they know what to do with these problems, and having a better appreciation of that advice would also stand us a long way.
Lillian, I see you have a heart up. Is that the same as your hand, or you can unmute yourself and ask a question if you want, or just you're agreeing with Dr. Bonomo's comment?
I want to give a shout out to Dr. Bonomo because our programs have collaborated and we have saved many lives thanks to him. And we're better stewards because of he's 24/7 available. And like he said, doing stewardship is an art, but your partnership has been crucial in helping us take care of people. So thank you.
Well, I'm a doc too, right? I don't want my patients to get sick on me. So.
Okay, well, I thank you. Any other questions? I thank everyone for joining us. Oh, I'm sorry. Someone from the ham oil. I don't know the name, so please unmute yourself and ask your question.
Dr. Wise, is Paula Lichtenberger from the VA. How are you?
Oh, Paula. Good.
So, Dr. Bonomo, hello from the Miami VA and from
the Cleveland VA. Yes.
I have a question for you, and it's just more a little bit of a comment. I struggle a lot as an antimicrobial stewardship, seeing this fight between the humans and bacteria, in which, every year I feel a little bit more hopeless in terms of I see that the bacteria are winning this war and this battle. And every time that I think about the battle between antimicrobials and bacteria, I start thinking about how to think out of the box. And how to think about things that are not antimicrobials that can help us out independently, despite of the infection control. And then I see here is like genetics and all the HEMON getting into very, genetic testing, genetic treatments. And how far are we in terms of trying to start getting into phages and effective CRISPR methods to really start fighting our battle against multi-drug resistant bacteria?
I think in the next couple of years, first of all, thank you. I think in the next couple of years, we're going to get there. I think we're going to get there. I think, initially a lot of us were a little suspicious of phages, right? And then all of a sudden you start hearing, these landmark studies. So, people are using it against abscesses, nice paper, Nature Micro there. People start using it clinically. Chip Schooley's paper, when he showed it, it worked against Acinetobacter. Phages are going to be like precision medicine. I don't think everybody's going to be able to do it. You're going to have to be really skilled. You're going to have to really know what you're doing. And you're going to have, the ability to, if resistance occurs to phages, which it does, we know that's going to be a fact. You're going to have something ready to go afterwards. But I think, the ARLG has a clinical trial that they're starting up on this, and I don't know where along it is yet. But I know Pranita Tama at Johns Hopkins, she's involved in one of these studies, and, I think we're going to see whether they work or not. A thing I do want to see from my own personal interest, I would like us to do more like, monoclonal antibodies against certain bacteria using monoclonal antibodies paired with antibiotics. I hope for that too, because I think we control that more than we do with phages. Phages, you're giving a living thing to a living person. You don't have that much control, but monoclonals you do. You can tailor a monoclonal specifically to a pathogen. We haven't capitalized on that as much, but, if you have, ID fellows in your program, have them think about, how can you make an antibody against Pseudomonas? How do you make an antibody? And people are doing it. They're trying their best. It's not easy, very hard to do, antibodies against bacteria, but I think they may have a role, particularly in acute sepsis, where you need to neutralize the organism immediately and stop it from being present. I think there may be a role for antibodies. And the last pitch, I think, I hope before I stop practicing, but I hope we could use immunotherapy too. How do we boost up our own immune system? Many of us, take care of, well, I'm over 65, no surprise. My immune system is not as good as it was when I was 35. How do we immunoadjuvant people? If they get acutely sick, how do we improve their immune response? In an acute. I think we're going to be in business a long time. We're going to be in business a long time.
Good, and be healthy and in business. That'll be great. So thank you very much again, Dr. Bonomo, for the grand rounds. I remind everyone to look in the chat for the link to the MOC and CME credits, and we'll look forward to seeing everybody next week. Stay healthy, stay well.